Hi class, and welcome to Chapter 9, Part 3 video notes. So let's talk a little bit about genetic traits and human pedigrees. So we kind of already discussed this a little bit in class, taking a look at that pedigree to determine the phenotypes of the family members and the inheritance of the trait through the generations. So through looking at the inheritance and looking at the phenotypes, we can then kind of be able to deduce the genotype of the family members. And as we saw, we can't find that for every family member, but we can for most. So this is going to also help us demonstrate dominant or recessive inheritance. The ones that we looked at together in class were examples of recessive inheritance. We haven't taken a look at dominant inheritance together in class yet. So a pedigree is going to show that inheritance through family, so it's kind of like a family tree of genetics. So here's a picture just showing you examples of some of the things that we can use pedigrees for. So freckles, widow's peak, or attached or detached earlobes. We also use it to take a look at diseases such as dwarfism, because that's a dominant trait, deafness, and other diseases such as Huntington's disease. So it's a very beneficial thing to have. So here's an example of a pedigree chart. So like you all did in class, we know the genotypes of everything that is filled in because they exhibit that trait. So we know that they're homozygous recessive. And then from there we worked backwards to determine the parents, grandparents, and siblings. And here's just another example. This is breaking it up, showing you the first generation parents, the second generation, and then the third generation. So once again, females are circles, males are squares. Affected individuals are going to be colored in. So now we're going to take a little bit of time talking about disorders. A lot of disorders in humans are controlled by one gene. So let's take a look. So inherited human disorders will show recessive inheritance and dominant inheritance. So this recessive inheritance, 2 recessive alleles are required to show the disease. So that's just like anything else that we've talked about thus far. So if it's recessive, in order to have that phenotype, there has to be 2 of the alleles. Heterozygous parents will not have the disease or show symptoms, but they could be carriers. So we talked about that in class a little bit already. They're carrying the allele of that disease. So they're going to be heterozygous. So we didn't talk about inbreeding at all in class, but the probability that you're going to have the disease will increase if your parents say were related, which for us it's gross to think about, but we didn't know these things, and back in the day, a lot of times kings and queens wanted to keep the bloodline pure. So what they did was they would marry off their children to first cousins. So that's going to increase the probability of getting the disease. For dominant inheritance, 1 dominant allele is needed to show the disease, just like other things that we've talked about so far. So if you have the dominant allele, whether it's in heterozygous or homozygous dominant form, you're going to show the disease. They're usually eliminated from the population because these people know they have the disease oftentimes, they won't have offspring, or they can pass away before it's time to reproduce. So they could die before reproductive age. So here's an example using the letter G. So the recessive disease is caused by having the 2 recessive alleles. So there's only a 25% chance that these 2 carriers, these 2 heterozygous parents, will have an affected child. So let's talk a little bit of some variation in Mendel's laws. So do a little bit of review here. Well, let's talk about incomplete dominance. So this is new to you because you're used to seeing dominant showing up if there's a dominant allele and the recessive phenotype only showing if there's 2 recessive alleles. But now it's a little bit different. We have something called incomplete dominance. So neither allele is going to be dominant over the other. So there's an expression of both alleles and there's an intermediate phenotype. So let's take that and break it down a little bit. Intermediate, it's like a middle version, and this phenotype is what it looks like. So there's like a middle version of what this child looks like in the heterozygous form. So it's kind of like blending. And the reason I say that is because in snapdragons, which are always fun to play with, snapdragons have an incomplete dominance when it comes to flower color. So a red parent mating with a white parent can produce all pink. So it's like blending, white and red making pink. So what we do to represent this is we use capital letters to represent color, and then there's like a little subscript to represent whether it's white, red, or pink. So we have our parent generation, our purebred red and our purebred white being crossed to have 100% F1 generation. Then of course we're going to cross this F1 generation with itself. So you see the genotypes of the parents, they're both pink, cross them with one another, and then by taking a look at this Punnett square here, you see that you now have a 50% chance of having a pink, a 25% chance of having red, and a 25% chance of having white. And of course you guys know I love this picture because they drew a little sperm tail, and so they actually showed them as gametes. So now we're going to move away from codominance, and we're going to move away from incomplete dominance, and talk about genes that have multiple alleles in the population. So multiple alleles, so more than 2 alleles are found in the population. So a diploid individual, such as you and I, can carry any 2 of these alleles. So something that's really popular to talk about in the scientific community with multiple alleles is ABO blood groups. Some of you may know your blood type, some of you may not, but it's very cool because there's 3 different alleles in the population. We have A, B, and O. So then this leads to 4 different varieties of blood type. We have type A, type B, type AB, and type O. So then that kind of leads us to codominance. With these multiple alleles in the population, neither allele is dominant over the other, and the expression of both alleles can be observed in the heterozygous individual. So a lot of you have heard the term AB blood. That's because A is not dominant and B is not dominant, so there's an expression of both alleles. This is called codominance. So we're going to talk about blood type a lot more in class. It's a lot easier to show you and teach you about this if I can actually draw a picture. So definitely read up on that. This is how we show these genotypes. This is a little bit different. If we're O, there's 2 lowercase i's. A, we use capital I's and then the subscript A. For B, capital I's, subscript B. So this is going to take a little bit of getting used to because you're used to using just one letter, lowercase, uppercase. So now let's talk a little bit about chromosome behavior and how that accounts for Mendel's law. So let's do a little bit of review. You guys talked about the law of segregation and how that works. So it depends on the separation of those homologous chromosomes in anaphase 1. So in metaphase 1, those homologous chromosomes line up together, and then in anaphase 1, they split apart. Those chromosomes could have aligned in any specific way. It could have been, depending on how many homologous chromosomes you have, there could be a lot of different ways they can align. But however they align is how they're going to pull apart during anaphase 1. The law of independent assortment depends on alternative orientations of chromosomes in metaphase 1. So this is what talks about the actual assortment of these chromosomes in metaphase 1. So let's take a look here. So in this picture, we start with an individual, and it's got all round yellow seeds, so it's heterozygous. So you can look here and see that there's a big R, big Y, little y. So meiosis, we're going to duplicate our chromosomes, our genetic information, and it shows their metaphase 1 of meiosis. So those chromosomes, those homologous pairs, are going to align at the equator, and they can align in any way. So it shows you these two ways here, how they can align at the equator. So then it shows them splitting apart in anaphase 1 of meiosis and the different ways that can happen. So then in metaphase 2 of meiosis, those sister chromatids align at the equator, which we've talked about a lot. So those sister chromatids align at the equator, and then when they pull apart in anaphase 2, this is what we have an end result once those cells will separate. So in anaphase 2, the sister chromatids pull apart, and then depending on which side they go to, depends on which gamete they are inside the different possibilities. So then we take a look at fertilization among the F1 plants. So here are our F1 plants here. All of our F1 plants. So what it's taking a look at is if these gametes were to come together, what the genotype would be. And this fertilization will give you the F2 generation, our 9:3 to 3:1 ratio. And we're going to talk about this a lot in class. This is kind of a hard concept to grasp, I feel like, until you get the hang of it. It takes quite a bit of practice talking about it and thinking about it. So, sex chromosomes and sex-linked genes. These are always really fun to learn about. So, chromosomes determine sex in many species. So we talked about this with humans. XX for humans is female and XY is male. And we also talked about how the male determines the sex of the baby. So, the XY system in mammals and fruit flies, that we just talked about, it's different in grasshoppers and cockroaches, such as XX and XO. For birds, butterflies, and fishes, they use Z and W, but we're just going to talk about humans. So here's the X, an actual X chromosome, and here's the Y. So there's a big difference. So sex-linked genes are the genes that are just located on the sex chromosomes, so either X or Y. Remember, autosomes are 1 through 22, and those are just, they're not involved in sex-linked genes because the sex-linked genes are either on the X or on the Y. So X-linked genes are passed from mother to son and mother to daughter. X-linked genes are passed from father to daughter, and Y-linked genes are passed from father to son. We know that makes sense because only the sons will have the Y gene. It'll only have the females will get an X from the father, and the sons will get a Y. So this is seen in fruit flies. So having depending on having a red or white eye is a sex-linked trait. So let's take a look at it really quickly. So the female and the male mate, and here's the possible sperm here, and then the offspring. So here's the red-eyed allele is capital R, and the white-eyed allele is lowercase r. So then the female and the male, their possible sperm. Notice here the white-eyed male, because they get that affected X chromosome from mom, from the egg over here from mom, and they don't have another X chromosome to compensate. They only have Y. So they're going to show that characteristic. And here again, here's the female, a female that's a carrier and a male that has white eyes. That's going to even increase the chances of having white eyes even more. Mom gives the recessive allele, and dad gives the recessive allele on his X, so half the females will have it and half the males. So there's also sex-linked disorders, and they affect mostly males. We'll talk about that a little bit more in class. So I hope you enjoyed.